All right, good morning, everybody. Okay, let me bring up my screen. And I believe that we ended up around here. Okay, so just to recap, we're basically starting at the beginning, at the big scale of the control of eukaryotic gene expression and controlling the access of the transcription machinery to DNA is a big part of that. And so we've already gone through this list. I'm not going to spend much time on it, but what we're going to do is go through these different modifications in turn, right? So the first thing we're going to talk about are the chromatin remodeling complexes, and then the chemical modifications that we can make to histones and DNA, which can alter transcription or the rate of transcription. And so basically, chromatin remodeling complexes are ones which change the how DNA is wrapped around histones, or even whether it is actually wrapped around histones. So we can rotate these histones, so we can shuffle them along, kind of like just moving them like down the strand a bit to expose these binding sites. We can remove them entirely, and we can do some other funky things to the histone DNA relationship. Right, and so we do that using ATP, right? So they're large protein complexes. Actually, I don't like the word aggregates there. Let's change that. Aggregates implies that they're not incorrect. Conformation, right? That's kind of a word we use for other things. But they're a bunch of proteins that form a complex, and that complex uses ATP to carry out its tasks, right? So it's an energy intensive process. So it has to be a very deliberate one. And essentially what we're doing is making regulatory sequences either more or less available to the proteins that bind to them. So transcription factors, transcriptional activated proteins, title box binding proteins, stuff like that. Right. So remember, RNA polymerase cannot itself bind DNA alone. It has to have other proteins to help it. Right. And those DNA binding sites can be covered up or exposed depending on the action of these chromatin remodeling complexes. So obviously, depending on what you do, right, you can positively or negatively regulate transcription. And again, so this isn't just to kind of stress the point, this is a really important one. This controls the access to the DNA of the transcription machinery, right? That's the key point. So it doesn't matter if the proteins that promote transcription of a particular gene are present in that cell, if they cannot get to the DNA to do their job, to bind to it. So you can see this is like another level of complexity essentially. And so this is one example of how CRCs work, right? So essentially you can see here we've got this multiple subunit protein complex, right? And what it does is it binds to, and actually I'm not 100% clear how it recognizes different sites. I don't know whether it's, and this is actually partly how complex it is and partly the fact it's very hard to know everything. But my guess it would be that those remodeling complexes recognize certain modifications on those histones, right? And so essentially it's a kind of a cue. That's an educated guess, right? Because it's kind of, in a sense, it can't be DNA specific, right? So anyway, this chromatin remodeling complex comes in, and basically with the aid of ATP, so this is a rotational deal. So if you imagine that like DNA is wrapped around the histones and a binding site is essentially tightly bound to that histone, right? So even though the DNA is there, right, it's kind of visible essentially, the transcriptional activator protein and the like can't actually come in and bind to it because it's stuck to something. And so now what we're going to do is we're going to rotate those histones around, and just like, you know, I was thinking of a tape on a cassette, but most of you probably don't know what a cassette is anymore, or a videotape, right? But just as you can wind like a bit of rope or thread around a bobbin, right? You can do the same. And if you do that, then the bit that was over that histone now is moved between two histone or two nucleosomes, essentially, right? And in doing so, and there's a very particular spacing here, which kind of plays into that as well, right? There's no kind of surprise that these two things are the distance apart that they are. So once those sites are now accessible, right? So this is not just, sorry, got a bit of a headache. I'm blown around the road this morning, kind of riding in. Once this bit of DNA is accessible, now there's going to be a transcriptional activator protein, some transcription factors, and the like, which are able to recognize and bind to that. Tadalbox binding site, Tadalbox binding protein binding site, which is kind of cumbersome, is also available, right? So the TBP can recognize that, and now RNA polymerase can be recruited. And RNA polymerase is actually very good. It's kind of like a snowplow in a sense, also something you might not be familiar with, right? But it's able to kind of go along and push these nucleosomes off of the DNA, right? So as soon as the RNA polymerase comes in, we're good. But the key is actually being able to recruit RNA polymerase in the first place. That's the real challenging part. And so by freeing up these two sites, now they're available for RNA polymerase come in and begin the process of transcription. So let's put up the chat box just in case anyone has any questions. Okay. Also, another thing we can do, rather than just shuffling these around and rotating them a bit, we can actually just remove them, right? So if we really want to expose DNA for transcription, and again, this is a matter of degree, right? Just as with everything that we've talked about. Now we can get a different remodeling complex coming. This one doesn't rotate the histones or just kind of shuffle them along a little bit. It actually kicks them out, right? So it removes the histone completely, or the histones. And now we have a completely bare piece of DNA, which is available for transcription factor binding and recruitment. Now, obviously, that's going to result in even higher levels of expression because no longer we don't have to worry about the histones even being there, right? So that's a different way of kind of thinking about it. And so these changes are not necessarily heritable, right? So this is actually part of the dynamic flux of chromatin in the cell, right? So these bits of DNA are constantly being kind of opened up and then shut down and opened up and shut down, or maybe they're just shut down completely and then they're opened up for a very specific task or point in time in development or, the organism's life. So this is part of the, everyday dynamic, heterochromatin to euchromatin to heterochromatin to euchromatin. Now, there are actually a bunch of epigenetic marks which are very much heritable, right? And these marks are typically in the histone proteins themselves and the DNA as well, right? So again, these aren't changes in the sequence of DNA or the sequence of the proteins, right? The polypeptide sequence. They're actually things that are added to or removed from those two molecules. And generally speaking, there are two different modifications that occur. One is methylation, which typically is associated with repression, but it depends on location. Well, in DNA, it's about repression. In histones, it depends, right? Depends on what you're methylating and where. And so essentially, what we mean, I'll get the chat up. So methylation is the addition of a CH3 group to either a histone, typically a lysine, residue on the histone tail, or a cytosine in DNA. Forgot to add that. Acetylation, if I can spell it right. That's addition of a slightly larger group or acetyl group to a histone. And again, that's typically lysine. And this is astonishingly complex. So essentially, what we're doing is we're modifying the properties of, in the case of histones, we're modifying the properties of the histone. And so here's a kind of a question to make you think. What's the negative? Ooh, dang it. All right, so shot myself in the foot there. What charge do you think histones, particularly their tails, must have for them to be attracted to DNA? Think about what charge does DNA have? All right, so histones have to be positive because DNA is negative, right? I'll just type that out. And you can't change the charge of DNA, essentially, right? Because it is what it is. It's a fundamental property of the backbone of DNA. So the thing you can change is the charge of the histones. And so depending on whether those histones are positive or neutral determines how tightly that DNA binds to those histones. Okay, so if one of the residues that we're modifying is a lysine. Can you guess what type of amino acid residue a lysine is? So if histones are either positive or neutral, and we're going to be modifying a lysine, what type of amino acid do you think lysine is? No, opposite of that. It's positive. So how nice. Why is lysine not look right now? Spelt it. Huh? It's always weird when you type stuff up. Sometimes it just doesn't look right. And so lysine essentially is a basic amino acid. And so essentially by modifying lysine, we change its charge. And that's how we change how tightly histones bind to DNA. Now that's a pretty major simplification because it kind of depends which lysine and where that is in DNA. And that's really just the case for acetylation. When we're talking about methylation, that's even more complex, right? So typically whenever you see, talk about epigenetics in the literature, they'll tell you what lysine, right? So there might be something like H3K4, and that's histone 3, lysine 4 is being modified, for example, right? And so it very much varies, but the overall kind of aim is that we're either going to be causing those histones to bind more tightly to DNA or more loosely, right? And we can turn, that on or off, we can modify it. There's lots of different kind of switches that we can use to control that property. And also by adding these tags, right? So these tags are inherited every round of DNA replication because you reuse 50% of those histones. But every one of these tags can serve as another purpose, right? So we can use that to recruit chromatin remodeling complexes. And there's a good example down here. We can also have an interaction between the methylation of DNA and the modification of histones, right? So it kind of goes back and forth, right, between the two. There's a lot of crosstalk from the tags that are on histones and the tags that are on DNA and opening and closing the access to DNA. And so basically you kind of go between these two different forms, right? So here's the kind of like a bigger view of the nucleosome, right? So the previous time we showed this, we didn't really show these tails, right? So you have all of these tails sticking out. These are available for modification. And so essentially you can go between two different forms. You can go between highly methylated and deacetylated histones, which are closed, kind of transcriptionally off, and demethylated and acetylated histones, which are form open chromatin, right, which is uncondensed and accessible. So let me just bring up the chat. We have you'll have to forgive my spelling. And then if you remove those methylated groups, so you demethylate them, and acetylate histones, you get open chromatin or euchromatin and transcription. And for acetyl groups, you actually have two types of group proteins. We have, histone, come on, acetylases. If I can spell them right. So by acetylating the lysines in these tails, that DNA kind of is less tightly bound to those histones. It's more available, right? So proteins can bind to that DNA more easily. And you have, come on, get you back again. Histone deacetylases. These are called HDACs. These are going to remove those acetyl groups from the tails. Those tails bind more tightly to the DNA. It's less available for transcription. And really, it's a constant interplay between those two. Histone acetylases, HAs, and histone deacetylases, or HDACs, constantly switching chromatin between these two forms. And you can see here, like this is basically the kind of mechanism by which that works, right? If these tails are no longer positively charged or as positively charged, DNA is still wrapped around the histone proteins, but it's held much less tightly, right? Essentially, you have a weaker attraction between the neutral histones, or less positive, and the negative DNA backbone, which is wrapped around it. How about that? Does that make sense? And these tags, essentially what you're building up, obviously they change over time, but these are also, every time you go through cell division, half of the histones are going to carry those tags. Okay, hopefully that all makes sense. And so basically, these tags are maintained, right? So even though you only kind of, in each cell only inherits 50% of the old histones, somehow those new histones which are made must be kind of basically programmed the way they were in the parent cell, in the mother cell. And so how that's done is actually very complex and poorly understood, right? And basically, essentially, you have not just your DNA code, right, which determines what proteins are produced, what they look like, and where and when those proteins are produced. You also have your histone code as well, which determines which of those possibilities are kind of accessible, right? What versions, what subset of those combinations of genes being expressed is possible in that given cell, right? And there's a very complex interplay between the histones being modified, the DNA itself, and other protein complexes which use that information to do different things. Now we also have DNA methylation, right? And so the idea here is that here's cytosine, right? Here's a hydrogen atom that can be replaced with a methyl group, with a CH3 group. Now this is something that's very common in promoter regions, right? And so one of the, and actually this is really interesting because some work that students I supervised did at a previous institution was about looking at tumor suppressors, right? So genes which are involved in suppressing uncontrolled cell division, right? Say those cell cycle checkpoints are breached and a cell is inappropriately going through cell division, right? So there are a bunch of proteins called tumor suppressors, right? Because of their role in suppressing that process, which recognize that and either shut down cell division or signal for those cells to be killed or to kill themselves, right? Via apoptosis. Now, this work found that these particular tumor suppressors, I think they were looking at one in particular, P70, I think it was. They're called, they have a bunch of different names. P53 is kind of one of the most famous ones. What they found was that in patients with tumors, the promoter regions for that particular tumor suppressor was heavily methylated. We don't actually know why, but there's a very strong correlation between methylation of tumor suppressor promoters and tumors. So essentially, somehow, I don't know whether it's the tumor itself that's doing this, right? So it's kind of tamping down on things which are not good for it, or if shutting down tumor suppressors leads to tumor formation. It wasn't exactly clear which of those. But one way in which tumors proceed is by, the shutting down of transcription of things which would normally suppress them, right? And so this is actually a kind of a real interesting facet of tumor biology, cancer biology. And essentially the reason is that when cytosines are modified in this way, they're not recognized as cytosines, not readily so at least. So if you have a specific DNA sequence in the promoter region, which is recognized by transcription factors and transcriptional activator proteins, and that sequence contains Cs, if those Cs are methylated, then those proteins can't recognize them. And so even though the DNA might be available, right? So it's euchromatin, it's not tightly bound to the histones, it doesn't matter because the transcriptional machinery is not going to recognize it. And also these methylated cytosines are now recognized by other proteins which act to repress transcription. And so rather than, even though the sequence hasn't changed, how that sequence appears to proteins does change. And so those repressors come in and typically prevent the binding of transcription factors, which would activate expression. So this is basically a whole process specifically in the promoters, right? It happens predominantly in the promoters of genes that will transcriptionally silence a gene, even if the signals to turn on that gene are present. And this always happens, or can happen rather, at this motif, right? So basically it's CG, right? A dinucleotide motif. So you can imagine it's just two bases. There's going to be plenty of these around in the genome. And quite often you have clusters of these CGs, which are called CPG islands. And the P is basically the P for phosphate, right? So it's cytosine, guanine, and then maybe another base or two, and then cytosine, guanine. You'll find a lot of these, right? So these are essentially kind of signals, right? Or other switches that can be flicked to turn on or off genes, right? So those methylations can be removed. And let's get, oh, we've got the definition there, but it doesn't hurt. We have DNA methyl transferases, DNMTs, equals methylation and silencing. Demethylases I don't know if there's a shortening for those or not. Called removing methyl groups and transcriptional activation. If the other conditions are present for transcription to occur, such as the presence of those transcription factors. So we have specific regions in DNA which are recognized by these proteins and are used as another way to control transcription by turning it on and off. And funnily enough as well, these motifs are what are used by, or these methylated cytosines are what's used by the mismatch DNA repair mechanism to determine which is the new DNA to repair versus the template DNA, right? So as we talked about a while back, you know, when you make your new strand, that strand is not methylated, right? The old strand is, and so that mark is essentially used to differentiate between old and new DNA. And obviously, after a while, those DNA methyltransferases will come along and methylate those new cytosines wherever they should be methylated, right? So that issue is already gone after a while. And so these also cause an interaction between both the DNA and the proteins, right? So these methyl groups will actually recruit other proteins which recognize them, and those will activate, again, subsequent proteins such as HDACs or histone deacetylases, right? If you remember, HDACs remove acetyl groups, which leads to transcriptional silencing, right? Because the DNA is going to be bound more tightly. And so we have this interplay between them where a methylated cytosine recruits a protein that recognizes it. That protein recruits another protein, which removes the methyl group of a lysine residue on the histone tail. And then potentially that can also recruit another protein, histone methyltransferase, right, which methylates that tail, which even more so suppresses transcription, right, and inactivates that chromatin. So there's a very complex interplay between all of these different things, which is not very well understood. And there's actually, here's another example. What example is this from? Oh, basically read bullets. That's funny. And so here we have histones bound to DNA. Again, here's the code that we're talking about. So this is talking about histone 3 and the lysine 9 residue, right? So there's a bunch of lysines in these tails. And so you can see how we've got a histone that's methylated, recruits a protein HP1 that then interacts with our histone methyltransferase, which may kind of reinforce that methylation, maybe on other histones in the nucleosome or nearby. But it also interacts with our DNA methyltransferase, which then methylates those cytosine residues, right? So these two examples kind of shows how this is bidirectional. We can either start with methylated DNA, which then leads to more transcriptionally closed chromatin, or we could start with transcriptionally closed chromatin, which then leads to transcriptionally silenced DNA through methylation, right? And so it's crazy, right? How complex this is, right? And how many kind of variations on this theme that you can have, right? So actually, to be honest, I never read papers on epigenetics because it just makes my head hurt, right? Whenever they talk about particular, it's like they're talking in code, which really they are, right? They're talking in the histone code. But if you don't know what that means, it's just like kind of blows your brains out, essentially. And it really kind of gets at how every region of every chromosome, of every cell, is going to have a particular chromatin conformation, which is either open or closed or somewhere in between. And that particular conformation for that particular region, for that particular chromosome for that particular cell, depends on the balance between methylation and acetylation of histones and methylation of DNA. So now you can see how it's very possible to shut down entire swathes of a chromosome. Because you just do these things, right? And that DNA is off, right? You can also see how if you screw up that in one way or another, now you can actually have fairly profound effects on gene expression and the biology of that cell or that tissue, right? And so it kind of just gets at like how astonishingly complex eukaryotes are in terms of controlling gene expression. Really non-trivial. And that's before we even get to the control of transcription, right? Which is also crazy complex, right? So this is the kind of like the basic view of control of transcription in eukaryotes. Essentially, so this arrow here is where transcription starts, and the gene is kind of goes that away to the right. And upstream of that transcription start site, sorry, you have a range of regions, actually, right? You will always have a core promoter. And the core promoter is, you could actually call it the minimal promoter as well, right? It's another way of talking about it. It's essentially the minimum region that is required for RNA polymerase and all the associated gump that needs to bind for transcriptions occur to actually physically bind. Right, so you can actually find this out for different promoters simply by cloning, you know, different size pieces of it in front of, say, a green fluorescent protein. And then you can see, well, you know, if it's that long, we get expression. If it's that long, we don't, right? So that's the minimum length, right? It's the core promoter. Now this will lead to, if nothing else is present, you know, just we have the Tata box and binding sites for potentially, you know, the odd transcriptional activated protein or something maybe. That will lead to what's called a basal level of transcription, right? So you will get transcription, right, as long as there's nothing interfering with that. But you're not going to get really high levels of transcription. You're also not going to get no transcription either, right? Because there's not going to be really any way of turning that off, right? So this is the core promoter. It's like the bare bones necessary to turn that gene on. Now, on top of that, you will have potential, and that can be, I don't know, it varies from gene to gene, but let's say 300 to 400 base pairs, right? They can be actually pretty small. Now, on top of that, you're going to have a whole bunch of regulatory elements, which can be really close by, right? So could be right next to the core promoter. So these are called cis regulatory elements. Let's just type that up. You don't see this in your PowerPoint? I'm sorry, Jelly. I did a little while back upload refreshed PowerPoints. This came up in the last section, and I'm pretty sure I uploaded a new version of this, but I will also, because I've moved some slides around just today, right, change a few words, I will upload a new version of this as soon as we finish talking. So you can use that if you want to go back over this. Yeah, good point. Thanks for that. I'm pretty sure I did do all of them for the rest of the course for both sections, but it's possible that I missed this one out. I don't think I did, but I'll upload it again anyway, and I'll let everybody know. So yeah, so essentially things are kind of like shuffled around as you kind of figure out how things go together. Well, these could be right next to the core promoter. And you can also have regulatory elements that are very far from the promoter. I don't actually know what those are called. I guess you call them trans-regulatory elements. I don't tend to use that phrase very much. And all of these, when I talk about regulatory elements, what I mean are sequences of DNA that are recognized by proteins, right? So those proteins may be transcriptional activator proteins, so proteins that promote transcription. They could be repressor proteins, so proteins that inhibit transcription via different mechanisms. And also ones which kind of enhance transcription, right? So they're not required for basal levels of transcription, but their presence will increase the level of transcription of that gene. And what you'll often find is that the DNA sequences for both promoters and repressors, or enhancers and repressors, often overlap. So if a enhancer or transcriptional activator or transcription factor is bound to one of these elements, it prevents a repressor from binding, and vice versa, right? So this is kind of one way in which you can tweak the level of transcription. And you can have many, 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 many of those DNA sequences, different numbers of them too, right? So the more copies of one you have, the higher the level of transcription you'll get in the presence of that protein, right? So now you can start seeing how complex this can also be too, right? So we have all kinds of other types of cis-regulatory elements, right? We have response elements, right? These are, again, sequences that are recognized, and these can allow like cell-specific or condition-specific transcription. We've got insulators, right? These are basically stretches of DNA that recognize proteins that prevent essentially the activation of one gene affecting the transcription of another gene. We've got silencers, which are bound by repressor proteins. We have enhancers, which are bound by activated proteins. All kinds of stuff. And essentially, just for one single gene, the output in terms of transcription is the sum of all of these, right? And this is really where we get our cell-specific expression from, or a large part of it at least. Really comes from which of these cis-regulatory elements, and particularly response elements, are present in the regulatory promoter of that gene, and which proteins that may or may not recognize them are present in that cell. And so you can see here, right, we've got, these are 3 different genes. This is actually a viral gene from a virus called SV40. And these are from a couple of other different genes. And so you can see, at least, you know, this one doesn't have a tartar box because it's viral, but you have this, you know, core promoter, right, which is a, you know, specific size and is really kind of a little truncated in these examples, but it's what is necessary for the binding of RNA polymerase. And then in the regulatory promoters, you can have a range of different response elements, range of different things that are recognized. And these are called, in this case, GC box and oct box, a count box, right? And how many of these you have and what you have can determine how highly expressed that gene is. So for example, the SV40 promoter drives a huge level of expression. This is actually a promoter that's used quite often in systems used to produce proteins, right? So if you want to produce a lot of protein, whatever that might be, insulin, let's say, you would put that under the control of an SV40 promoter, right? Because that will just drive really high levels of expression. Now, maybe that's not such a good idea because high levels of protein production might cause problems, right? Aggregates and things like that. And so you'd use something different. And so each individual gene that we have consists or has a different combination of elements in its regulatory promoter. So, you know, this one, for example, may be kind of more context specific. Histones, you're always going to need histones, but you need more of them during DNA replication. So you may have a different expression profile from this combination than you would from this one. This may be expressed in all cells and at, you know, a kind of a fairly constant level going up during DNA replication in cell division. This gene may be more cell specific, right? So it's only going to be expressed where you have all 3 of these proteins that recognize these 3 different response elements, which may only occur in a particular subset of cells. Does anybody have any questions? Actually, pretty much done for time. So I think this would be a good place to pause. And on Friday, we will kind of continue going through. I've got to dig out some examples of all of this jazz because we've got quite a little bit left to do. I'm going to talk about alternative promoters, different combinations of things which drive expression, kind of gene structure stuff, and also some alternative splicing. I've got an actual example from my own work that we can use for that. All right. Well, thanks for stopping by, everybody. I shall see some of you in an hour and a bit, and the rest of you I will see on Friday. So thanks again, and have a great rest of your day.